Welcome to today's webinar. My name is Jose Francisco, Project Manager at the IAS-USA. This webinar is the third in the four-part webinar series that goes over updates from CROI 2020. We are excited to have today's presenter, Dr. Katharine J. from the University of Pennsylvania, who will discuss data presented at CROI regarding HIV cure research. Welcome, Dr. Barr.
Thank you. All right, it's really a privilege to be here and be able to talk about HIV cure research. I'm sure many of you have been consumed with the COVID-19 response, and so it's a nice break from that to be able to discuss the exciting research that happened at CROI. And I think about CROI as kind of an interesting kind of marker of when, for me, the epidemic really took hold, because it was transitioned from an in-person meeting to a virtual meeting. And I have been very busy since CROI and maybe haven't had as much of a chance to scroll through all the webinar and online materials that are available. So one of my goals for today is to try to highlight some of the seminars and some of the oral abstract presentations that I thought were particularly interesting or relevant. And clearly there are many more that I'm not able to cover, but hopefully this will pique your interest and you can go to the website and look through whatever websites, whatever webinars and talks you find most interesting. So with that, we'll go through some of the CME information. And I'm just going to briefly scroll through these, mentioning that you can get CME information as physicians, as well as via nursing and pharmacy credits. Grant support for this webinar was provided by these supporters, and we're very thankful for this opportunity. As far as navigating the webinar, if you have any questions, in terms of first the poll questions, a separate window will open to show the poll, and you can choose your response within that poll, and then the responses will be displayed after the poll closes. If you have any specific questions, you can type them into the chat box. And depending on sort of how flustered I am, we'll either deal with those during the webinar or at the end during the question and answer session will be collated by our IAS folks. So the first poll is, from which geographic region are you viewing this webinar? Okay, so we have people from all over the world, which is very exciting. We have some people from Europe and Africa, as well as people who are here earlier in the West Coast all the way through the Northeast. Fabulous. Okay. So hopefully we all got a sense of how to use the poll. Poll number 2. This one is slightly more relevant. Have you accessed any of the resources, either electronic posters, webcasts, abstract ebooks, mobile app, etc., from CROI 2020? And just as a public service announcement, if you have not accessed these resources, they are open and available to both registered attendees and everybody else in the world. So please feel free to do so. All right, it looks like about half of the participants to this webinar have already accessed information from the website, and I hope that this inspires additional folks to do so. Okay, so to access such materials, you can go to this website and navigate from there. Okay. And it is a slightly new website, and there's more information on the website about that. So we just want to mention there's some other webinars coming up that may be of interest to this audience. First, on April 29th, which is tomorrow, there's going to be a well-being during COVID-19 crisis for healthcare practitioners. This is particularly relevant for this audience, I think, and I encourage us all to consider watching this and consider this topic. And then the next webinar in the series of highlights from CROI will be a highlights in clinical research by Paul Sachs on Tuesday, May 5th. And you can register for that the same way you registered for this. Okay, now to begin our update of HIV cure research. And thank you again for your attendance. My name is Katie Barr. I'm an assistant professor at the University of Pennsylvania. So the learning objectives for this particular webinar are that hopefully we will be able to summarize recent developments in the current understanding of the latent reservoir, identify recent cure strategies that are being tested in preclinical models, as well as promising strategies that have been moving into clinical trials. Our pre-test question number 1. Okay. As the intact proviral DNA assay, or the IPDA, has been applied to cohorts of ART-suppressed people living with HIV, we have learned, 1, that CD4 T cells with intact and defective provirus decline at the same rate, B, CD4 T cells with intact provirus decline faster than those with defective provirus, or C, CD4 T cells with intact provirus decline slower than those with defective provirus. Okay, great. It looks like we have a option B with a slight advantage, and hopefully the answer to this question will be clearer as we go through the webinar. All right. Our next pretest question. Which of the following statements is false about latency reversal agents, or LRAs? A, HDAC inhibitors tested in human clinical trials are generally safe but do not lead to increased plasma viremia or decreases in the reservoir. Again, which is false. B, non-canonical NF-kappa B activators demonstrate both latency reversal, meaning plasma viremia, and reservoir reduction in ART-suppressed non-human primate studies. Or C, TLR7 agonists in combination with broadly neutralizing antibodies have shown delays in rebound after treatment interruption in ART-suppressed non-human primate studies. This is a tough one. Okay, so it looks like we're pretty evenly split across all 3 options, which hopefully will improve after the webinar, and we will address this as we move along. Okay. All right, now to begin this discussion. So I like to discuss, or to begin cure discussions in general with the question, why are we as a scientific community even pursuing cure? We know that antiretroviral therapy is highly effective in suppressing viremia and reversing or preventing immunodeficiency. However, we also know that the stigma of ongoing HIV, of living with HIV, the cost of treatments, issues with access to these treatments, and potential toxicities, and the requirement for, at least at this current time, daily adherence. All these things together are drawbacks to our current antiretroviral therapy. But I think most importantly, it's really key to recognize that the community of people living with HIV very much supports that we pursue cure strategies and we pursue sort of remission or functional cure strategies. So I want to highlight first a plenary from Dr. Sharon Lewin, who really discussed this issue as well as many others. So her first morning plenary was about HIV cure from bench to bedside and to community. And she really emphasized the importance of engagement with the community at all levels of the HIV cure development process, saying we need to continually work with people living with HIV to understand their preferred characteristics in an HIV cure and ensure that we are thinking now about how a product will ultimately be delivered in the field. So there was a lot of wonderful information conveyed in this talk, but I think this message of engaging with the community at all steps is really highly relevant. Okay, another thing I like to start HIV cure discussions with is almost kind of a disclaimer, because there's a lot of contention about what we should be referring to our goals here. There are a lot of ways of thinking about cure. Two general broad categories are either eradicative or functional. So an eradicative cure would be one where we completely eliminate cells bearing replication-competent HIV. It's estimated that this is going to take a serious effort with many logs of reduction in the reservoir and is a very high bar. But it is still certainly a goal. The other strategy is referred to as more of a functional cure, and this really exists on a continuum where we would have either complete eradication or eradication of some portion of the reservoir coupled with ongoing suppression by various immune or other surveillance strategies. And so I think these definitions are important as we're discussing both eradicative and functional cures in our general cure or remission strategies. So when we think about cure, we recognize that the fundamental barrier to cure is the persistent latent reservoir. And the figure I'm showing here is a study that was published in 1999, a long time ago, where people living with HIV who were suppressed on antiretrovirals had their treatment interrupted, and lo and behold, virus returned and rebounded in the vast majority of those individuals very quickly. And so studies like this have been the unfortunate sort of proof that there is this reservoir of HIV cells, HIV-infected cells, that is always there in our patients who have even very well suppressed HIV. So the definition of this reservoir is a replication-competent HIV provirus integrated into a long-lived quiescent cell that persists despite suppressive antiretrovirals. And although we've known about this reservoir for many years, decades even, there are still fundamental questions that remain unanswered. Some of those key questions are when the reservoir is formed, what cells and tissues are most relevant for the persistence of this reservoir, how is the reservoir maintained, and what is the role of expanded clones within that reservoir, and how can we best measure this reservoir? And so I think there were a few interesting updates to these basic questions that were discussed at CROI, and I want to highlight some of them now. One of the first things that I think was addressed in really an interesting study was the issue of clonal expansion, or expanded clones within the reservoir. So since about 2014, there have been several groups identifying the fact that our HIV reservoir has a large number of identical cells with identically integrated provirus, or cells with identically integrated provirus. So these expanded clones were largely thought to be bearing defective virus initially. However, in the past few years, it's become more and more clear that a lot of our replication-competent virus is also a part of these expanded clones. And in fact, recent studies have shown that the replication reservoir, in fact, has, in many instances, more than half of its virus as a part of these expanded clones. This is a study from the Silicano group from a year or two ago, where they basically showed that the composition of these clones waxed and waned, or increased and decreased over time. So there are mechanisms by which this dynamic process of clonal expansion and contraction occurs. And one of the things the field has been very interested in is what are the mechanisms driving clonal expansion and contraction. And three specific mechanisms have been suggested, including, see if I can get my arrow going here. Maybe not. Well, including antigen-specific expansion, whereby some chronic antigen is stimulating a certain cell type to persist. There's also homeostatic proliferation, where the natural sort of processes of immune memory via various cytokines lead to expansion of these cells. Or perhaps virus integrates at a specific part in the genome that leads to expansion of that clone. Well, one of the studies that was presented in CROI this year, an oral abstract by Francesco Simonetti, really dug into the specifics of antigen-driven clonal selection. So I wanted to review some of his findings. So what he did in his group was they took 10 participants, people living with HIV, and they did basically virus outgrowth assays after stimulating the cells to specifically have certain antigen-specific cells grow out. And so what they looked at was 4 different categories of cells. Cells that had no antigen stimulation, cells that were non-specifically stimulated, as shown here in kind of gold color, cells that were stimulated by CMV, and cells that were stimulated by HIV gag peptides. And what they showed in general is that when they looked at, and you can see these phylogenetic trees in the middle, when they looked at the non-specifically stimulated cells, you can see that these black dots are well distributed throughout this phylogenetic tree, and there are not that many identical sequences. However, when you look here under the CMV-specific phylogenetic tree, you can see that there's many different identical sequences. For instance, these gold clusters showing that these CMV-specific cells are highly expanded into these identical clonal populations. Another thing they looked at over here in this comparison in the figure on the far right is just looking at the the clonal identity of the cells on the y-axis and the clonal identity of the cells, a virus on the x-axis, and you can see that these are highly correlated. In the CMV-specific cells being much more clonal, both in terms of the identity of the virus and the TCR. So the next thing they did, which I think is fascinating, is they looked at what fraction of these CMV-specific clones contained HIV provirus. And what they found was this was really variable. So there were some very large CMV-specific clones that had only a small fraction of their cells bearing HIV, and there were others where the entire population almost had HIV infection. And this suggests 2 different time courses, both which were detected. One was that a clonal population was expanded maybe by CMV stimulation or seeing that antigen, and then at some point in the life of that clone, some of that clonal population was infected by HIV and went on to persist in that clonal state. In the other situation, you have a cell that is infected by HIV and then expands to become a clonal population. So I think that the take-home message from this really lovely study is that antigen-specific stimulation is a mechanism that drives clonal expansion of our HIV reservoir. Sometimes this happens with the whole clone being HIV positive. Sometimes it happens in only a portion of the clone. And also in this study, they were able to detect replication-competent HIV within some of these clones. So this is a relevant mechanism for the persistence of the reservoir. Okay, so next on to measuring the latent reservoir, which is a huge challenge because many of our studies have been discrepant. So the fundamental challenges are that replication-competent latently infected cells are very rare. They're spread throughout the body. They're silent or quiet, and they're outnumbered by cells bearing defective viruses. So they're essentially very difficult to distinguish from each other. But measurement of this reservoir is really key, because if we cannot measure this thing, it's very difficult for us to know whether any of our strategies are effectively utilizing it. And so instead of thinking a needle in a haystack, I think it's apt to think of a needle in a haystack, because many of these cells that we're trying to distinguish from each other look very identical in many of the ways that we're looking for them. So one of the approaches that's been used to characterize the reservoir that was presented at CROI was the use of whole body PET imaging. So this method was published a few years ago by Phil Santangelo and colleagues in a Nature Methods presentation. And basically what they used was radio-labeled anti-HIV antibodies, and they gave them to both viremic and ART-suppressed rhesus macaques that were HIV infected. And what they were able to show with this lovely technique is that ART-suppressed rhesus macaques had uptake in multiple different sites that we think are highly relevant for the reservoir, in the colon and the associated mucosal tissues, lymph nodes, the small bowel, nasal turbinates, as well as the genital tract and the lung. And so this was a description of how this study could be used in an animal model. So one of the abstracts that was presented by Tim Henrich was applying this strategy to human cohorts. And I think this is potentially a really breakthrough technology. So what he presented was a study looking, initially a pilot study looking in 15 people living with HIV. 5 individuals were viremic, 5 were suppressed on antiretrovirals, and then there were 5 HIV-negative controls. They were all given zirconium-labeled VRCL1, which is an antibody to HIV. This zirconium has a long half-life, which enabled longitudinal imaging over a period of several days. And what I'm showing here is a cut from their presentation where they're showing several lymph nodes in viremic individuals as well as ART-suppressed individuals compared to uninfected individuals. And importantly, what they showed in the ART-suppressed people living with HIV is that, again, we saw uptake in tissues we think are really important for the reservoir, in the nasopharynx, in select lymph nodes, as well as the gut and the bone marrow. They were able to correlate this PET imaging with actual biopsies where they measured P24 positive, CD4 positive T cells, and what they showed in this figure on the right was a very nice correlation between their sort of in vitro tissue measures and the imaging study. So Tim ended his study by discussing a novel improvement in this strategy, this quote, explorer high sensitivity PET scanning. And what I'm showing here on the left is an explorer image versus the traditional PET MR. And what you can see is that we've really enhanced, according to the researchers, enhanced sort of the sensitivity and the specificity by cutting down on background noise. And so I think these strategies are really promising modalities for us to understand and potentially monitor HIV reservoirs moving forward. So in terms of measuring HIV reservoirs, one of the important issues has been how do we do this in vitro in our assays, not just with imaging. And this is a study from a long time ago, 2013, where multiple labs contributed samples or contributed assays to the same samples. And what you can see is that many different assays get a wide range of values, and in general there was poor correlation between the assays. And this is because we have demonstrated time and time again that the vast majority of integrated virus is defective. And if you are measuring defective virus with one method and you're measuring only intact virus in another method, you're going to end up with very different answers. And so PCR-based assays with primers in conserved versus unconserved areas will lead to very different results. And our standard of care in many of these instances has been virus outgrowth assay, where we take cells and we stimulate them and we see which of those cells can produce replication-competent virus. But what we know about this assay over the past 5 years or so is that this substantially underestimates the virus reservoir, and there's actually a large population of virus that appears to be intact and inducible, but is not, for whatever reason, induced in our traditional assays. So one of the assays that's come online, as well as several similar assays, but I'll just focus on this one for today, is the intact proviral DNA assay. And this was developed by the Silicano group and Greg Laird. And what it does is it's a digital droplet PCR assay where probes are placed in strategic places throughout the genome where you can pick up and distinguish between intact or nearly intact virus and defective virus. And so this high throughput assay allows a readout of the quantity of both intact and defective proviruses. So in the initial publication by Katie Bruner, which was published in Nature in 2019, they demonstrated some really impressive qualities. They're able to, with this assay, exclude 97% of defective viruses. And in fact, 70% of the viruses identified as intact were borne out to be intact by near full-length sequences. Importantly, there's a strong correlation with our virus outgrowth assay, which is reassuring because previously we had seen very poor correlations between assays. And it tends to be about 100-fold higher in the IPDA than that in QBOA. And what they showed initially in the right here is that if you look at people living with HIV on suppressive antiretroviral over time, in general, they saw a decline in the number of cells with intact virus that was greater than cells with defective virus. So that was an initial hint at what we could do measuring cohorts or studies over time. So since then, there have been multiple applications of the IPDA to cohorts of individuals, and I think this is an exciting area. So Paluzzo et al. In JCI Insight applied the IPDA to the SCOPE cohort, and this was just recently published. They looked at 81 ART-suppressed people living with HIV who were studied over a median of 7 years. This SCOPE cohort is largely male with a median age of about 47. And what they showed in this interesting description of this cohort is that using the IPDA assay, in fact, intact proviruses declined in frequency more than defective proviruses. The initial, as you can see from this red line describing the slope, they fitted it to a model showing a two-phase slope in that early decline was more steep than later decline, with the early decline having these cells having a half-life of about 4 years, whereas after about 7 years, the decline slowed down quite a bit to about 18 or 19 years. And again, they correlated faster decay rates with higher nadir CD4. So the more intact your immune system is, when you start antiretrovirals, that was associated with a greater decrease in your replication competent or your intact proviral DNA compartment. So at CROI this year, there was another oral abstract that was presented applying the IPDA to a cohort of people living with HIV on antiretrovirals. So in this abstract presented by Raj Gandhi, they looked at a cohort from the ACTG, A5321, or the ARC cohort, and they tested 50 participants who had samples approximately 4 years apart. These individuals had been on antiretrovirals for a median of 7 years. And they looked at total DNA, cell-associated RNA, and residual viremia, the sort of measures that we had been using prior to the IPDA assay. And what they showed was no real change over time. However, when they applied IPDA to these things, or to these samples, what they showed was there was, in fact, a decrease in the levels of intact proviral DNA over 4 years, but there was not a decrease in defective or total DNA. And again, the half-life over this period of time for the intact PDA was about 7 years. So there is actual decrease in this compartment. Again, the defective proviral DNA and total DNA did not appear to decline very much. And so I think the take-home point from these studies is that this assay, which is high throughput and allows some transparency and follow-up into its methodologies, allows measurement of intact or sort of a more relevant reservoir measurement over time. And I think the use of this in clinical trials and in clinical cohorts is just going to increase in the next few years. Okay, so let's move on to cure strategies. So I want to highlight a talk given by Nicolas Chamont during the new investigators workshop that's done on the Sunday prior, the day prior to the, or the day of the opening ceremonies. So I would say, in addition, all of the new investigator workshops are really excellent introductions and even deep dives into a range of really relevant fields. But Nicolas spoke to us about cure strategies, and this is a slide stolen from his presentation where he talks about some of the different strategies. And if any of you attended this webinar last year, I spent a lot of time talking about strategies to limit the establishment of the reservoir via early initiation of antiretrovirals. And I'm not going to talk about that very much, but that is certainly one strategy that is being used to reduce reservoirs. There's also the attempt to reduce the size of the reservoir after individuals have started antiretrovirals. And there's multiple different approaches to this sort of middle section. And finally, there is the effort to suppress or to control the reservoir through immunotherapy via vaccine or other immune mechanisms. And I think more and more there's an understanding that we would like to combine both the reduction of the reservoir to enhanced suppression or control of the reservoir for our combination immunotherapy strategies. So again, to this immune therapy shock and kill type approach, the components of this that we would like are first this shock component where we have latency reversal, or we take cells not expressing HIV, not expressing RNA or the full virus, and we induce them to show themselves to the immune system. Then we would like some sort of kill or clearance, and that's either by an improved host immune system or some exogenous or engineered cell. And then while this is going on, we want to make sure that we can maintain suppression of any virus that's released through this sort of shock process. So the first component of this shock and kill is the shock, or the latency reversal agents. And so again, this is the goal, to take a latently infected cell, which has little or no RNA expression or antigen presentation, and to make itself visible to the immune system. And this is by inducing RNA expression or protein presentation, or to just give, actually make virus present in the plasma, even while people are on antiretrovirals. We'd like to do this in a safe way without generalized T-cell activation, but it needs to be in a sufficient proportion and of a sufficient duration that we can pair this with killing strategies to actually make a measurable dent into the reservoir, which is something that is currently lacking. So there are many LRAs that have been studied, but largely one of the groups that has been most tested are these sort of first-generation HDAC inhibitors. And these are histone deacetylase inhibitors. And many of these, including vorinostat and panobinostat and romidepsin, have been tested in preclinical and in early clinical trials. And in general, these things have been found to be safe, to show some level of RNA expression, variable levels of viremia, but they have not yet been able to show a change in the reservoir or a delay in time to rebound. But these are the first LRAs to be tested either alone or in combination in human clinical trials, and I'm going to go over some of those data now. So one of the first trials looking at combination immunotherapy with these first-generation LRAs is the RIVER trial. And this is really kind of a milestone of one of our first combination immunotherapy trials. So this was published by Sarah Fiedler, in Lancet in March 2020. And this is a phase 2 randomized controlled trial of 60 male people living with HIV who were started antiretrovirals during early infection. And what they tested was antiretrovirals alone versus antiretrovirals in combination with this LRA vorinostat plus a chimp adenovirus therapeutic vaccine. Their primary endpoint in this study was to look at the change in total DNA at week 16. And what I'm showing here in the figure is comparison of the ART only, sort of in the lighter teal, and the ART plus this LRA plus vaccine intervention in the darker teal color. And unfortunately, what was seen is that over the various time points before, through the trial, and at their 16-week endpoint, there was really no change in the levels of total DNA. And so this was kind of a negative study in that regard, but this was safe, and it was an impressive ability to accomplish this sort of cure trial in this population. All right, so at CROI, there was another presentation of one of these combination strategies using an HDAC inhibitor. I apologize, somehow I've lost the notification, but this abstract was presented by Dr. Ole Sogard from Denmark. And this is the roadmap study where they compared the use of romidepsin with or without a broadly neutralizing antibody, 3BNC117, in individuals who had been on long-term antiretrovirals. So as you can see from the first schema here, which is a little blurry, basically they had two groups. of 10 participants each. In the first group, you got 2 cycles of the LRA, romidepsin, and in the second group, B, you got both the LRA plus a broadly neutralizing antibody. In the first figure in the middle here, what you can see is there was a small number of participants in each group, red and blue, who showed some plasma viremia above this 20 copies per mil threshold, but generally there was not a lot of observed latency reversal activity as measured by plasma viremia. And then when they went on to look at measures of the reservoir on the top here, looking at total HIV DNA from baseline to after the first treatment cycle and after the second treatment cycle, unfortunately they did not see any changes in the size of the reservoir. And then shown in the bottom on the right, after they interrupted treatment, what you can see is the vast majority of participants had viral rebound within the first 2 to 3 weeks. And this is very similar to what historical controls have been shown to do in terms of the timing to virus rebound after treatment interruption with no intervention. So this strategy was shown to be safe and have minimal serious adverse events. However, there was not consistent latency reversal, and there was not a measured change in the reservoir by total DNA, nor a change in the time to rebound. So I want to highlight another talk that sort of addresses latency reversal agents, and this is by Dr. Ann Sharuti from Emory and Yerkes, and she gave a symposium which I thought was really excellent, summarizing some of the exciting new LRAs that are now, we now have in vitro and preclinical data and are entering into human clinical trials. So the way she framed this issue is we sort of have our first generation of LRAs, which were really targeting gene expression in a general sense, and these have largely had minimal impact in clinical trials. This new generation of LRAs includes agents that are more specifically targeted to CD4 positive T cells, where we think the majority of the reservoir resides. The specific agents that she mentioned are TLR agonists, NF-kappa B activators, IL-15 superagonists, and then CD8 depletion. I'm going to walk through some of her findings, but this is relevant to a lot of the immunotherapy that was presented at CROI. So Anne went on to talk about one of the more exciting LRAs, and this is the non-canonical NF-kappa B activator, or sometimes referred to as a SMEC mimetic, AZD5582. So this particular LRA has been recently published in Nature 2019 by Nixon et al. And what they showed on this figure on the left is that in SIV-infected macaques who had been suppressed on antiretrovirals for a long period of time, multiple doses of this LRA, AZD5582, were able to show viral rebound in the hundreds to thousands of copies per mil in these animals. New data that Anne showed is shown on the right, and that they were able to do a second cycle of this LRA, and they again saw consistent viremia, so latency reversal, in this later group of after the second cycle. So this group, excitingly, or this agent showed both reproducible and sustained LRA activity in multiple non-human primate models. She also briefly addressed TLR7 agonists, which also have been shown to have latency reversal activity in rhesus macaques. But her thought process was that sometimes the TLR7 agonists in some models have been less consistent or less high level or sustained in their LRA activity. But importantly, even these most exciting LRA activities or studies in these rhesus macaques have shown consistent latency reversal in terms of plasma viremia, but none of them has been able to show a delay or change in the size of the reservoir or a delay in the time to rebound by latency reversal alone. So the other strategy that she mentioned was pairing these LRA agents with CD8 depletion, or thinking about CD8 depletion as a latency reversal strategy on its own. So this strategy was first published in January of 2020. And on the left is data from that study where basically the IL-15 superagonist N803 was given to animals, and by itself these ART-suppressed animals did not show a lot of virus reactivation as shown in group 1 N803 alone. But in group 3, CD8 depletion plus N803, you can see there's a high level. Many of these animals have high levels of virus reactivation and plasma that's virus detected in the thousands to almost tens of thousands of copies. What she showed today at her lecture at CROI this year was additional data looking at a subset of these animals who had the lowest plasma viremia on antiretrovirals. So these were animals who had low virus, less than 3 copies per mil, and that's really analogous to what we think the majority of people living with HIV on long-term suppressive are, the level of viremia in their background. And for these these animals, sort of the hardest to show latency reversal, they also showed consistent and moderate level latency reversal with CD8 depletion and N803 in combination. So this CD8 depletion also was paired with a non-canonical NF-kappa B activator and increased the number of rhesus macaques that responded as well as the frequency of the response rate. And so there's optimism that these strategies paired together might be able to show similar latency reversal in human trials. So moving on to some of the trials that were presented in CROI using these new LRAs or other combination immunotherapy strategies. So I'm starting as background with a study published by Borducci et al. In Nature in 2018, which looked at the combination of broadly neutralizing antibodies and a TLR7 agonist. And so in this study, rhesus macaques, a total of 55, so a sizable number, were infected with a SHIV, and 7 days, or just 1 week after infection, they were started on antiretrovirals and maintained for 2 years. And then they were given a series of bNAb plus or minus TLR7 antibody antigen treatments, or agonist treatments, and then allowed for these things to wash out, and then we had treatment interruption. And what you can see in the bottom right-hand graph or figure is that the PGT121 plus the TLR7 agonist had a substantial reduction in the number of rhesus macaques with either delayed or prevented rebound. And in fact, several of these rhesus macaques had no rebound at all, and in very stringent adoptive transfer experiments were shown to have substantially reduced numbers or amounts of the reservoir. So this is the baseline and the promise of these TLR7 plus bNAb strategy that sort of we had coming into CROI. So one of the studies that I think was interesting was presented by Denise Xu and her colleagues from AFRMS in Thailand, where at their primate center, they tested a very similar strategy with a few modifications. So in this study, they had rhesus macaques that were infected with a clade C, a slightly different SHIV, and they started antiretrovirals at 2 weeks compared to just 1 week of antiretrovirals, which I think is a substantial difference. They had a shorter period of antiretrovirals pre-intervention, and they gave 2 bNAbs as opposed to 1. And so what you can see in the middle figure here is sort of a Kaplan-Meier curve showing the time to viral rebound. And what you can see in the blue line, which indicates the animals with active treatment compared to controls, is there was a delay in the time to rebound, a median of 6 versus 3 weeks. However, they saw less substantial effects in terms of the ability of these animals to control viremia post-rebound. And so I think that this is important for a couple reasons. First, it furthers the promise of this TLR7 plus bNAb strategy in modifying or delaying the rebound. But the effects were less dramatic. And this is probably because of the changes in this non-human primate experiment where antiretrovirals were started later. So there was a lot more virus replication and therefore a larger reservoir to contend with, potentially more immune damage from 2 weeks versus 1 week of infection. But this is the sort of model that may be more translatable into human populations where we rarely start antiretrovirals prior to 2 weeks after treatment interruption. Okay, so another study working within the TLR7 agonist realm was presented by Dan Baruch, and he and his colleagues did another large study looking at the effects of combination immune therapy in rhesus macaques. So what they did in this study was give a combination of a therapeutic vaccine, an AD26 plus MVA approach, with this TLR7 agonist plus bNAbs. And on the left here is a Kaplan-Meier curve showing when these animals rebounded. And what you can see is that the animals who received the antibody plus the TLR7 agonist in red and blue had a delay in rebound compared to the animals that did not give the TLR7 and the antibodies. In the middle figure here, we're showing the post-ATI viral set point. And importantly, if you look at the animals that got the therapeutic vaccine shown in the second and the fourth group, the VAC plus the VAC plus antibody, you can see that their viral load set point was a bit lower than the animals who did not receive the vaccine. So you can see these two different strategies were effective or suggested results in either delaying rebound or leading to post-virus suppression. Importantly, this study also showed that either the timing of antiretrovirals or the viral load prior to antiretrovirals was important in how effective the delay in time to rebound was. So on this final graph, we're seeing that animals that didn't rebound on the right side compared to those that did rebound, the animals that showed a more substantial effect had, with suppressed rebound, had lower pre-ART SHIV viral load, suggesting that how we design these studies in terms of ART initiation has a lot to do with which endpoints we'll meet. Okay, another immunotherapy, combination immunotherapy with BNAB study in non-human primates that was presented at CROI was presented by James Whitney, also from the Boston groups. And so what they looked at here was two pilot studies of an immune modulator, IL-15 superagonist, plus antibodies, either one or two broadly neutralizing antibodies in non-human primates. And so in the first figure here, I'm showing that in animals that received this IL-15 superagonist, you saw an increase in cycling in key effector populations. And so they did see a biologic and immunologic effect. And then when you compare the groups who got the full range of this LRA/immune modulator plus antibodies compared to controls, you can see in red we have at treatment interruption a less high peak viral load and a greater level of suppression of virus compared to our controls shown in the black line. So again, this is another immunotherapeutic strategy that paired with broadly neutralizing antibodies suggests some enhanced immune-mediated control after treatment interruption. So one other strategy to mention that I think is, has a lot of excitement in the immunotherapy realm is the concept of checkpoint inhibitors. And these are a big deal in the oncology world, but in terms of HIV, there are multiple sort of mechanisms that are biologically plausible for why these things could be really important. So these checkpoint inhibitors, PD-1 and CTLA-4 in particular, are important regulators of cytolytic activity, and they also mark cellular exhaustion and immune dysfunction. And the thought is blocking this cascade could potentially have both latency reversal effects as well as enhance our function of our exhausted effector cells like CD8 T cells. These are widely used right now in oncology with indications both in melanoma and non-small cell lung cancer. But an early trial of these agents in HIV-positive individuals was actually showed biologic activity, but was stopped early because of concerns with immune-mediated toxicities like hypophysitis. So this early trial highlighted the risks and the benefits of the current formulations of these agents in our very, in general, healthy people living with HIV as compared to those with serious or life-threatening cancers. So I just want to highlight there was another study published recently, just this month in Nature Medicine by Mirko Perdini and Justin Harper, where they showed sort of that combination checkpoint inhibitors, PD-1 and CTLA-4 blockade together, had a pretty substantial effect in rhesus macaques. So they looked at a very long and complicated study of SIV-infected ART-suppressed rhesus macaques, and these animals were given either PD-1 or CTLA-4 or combinations or a combination antibody. They demonstrated a bioactivity in the blood and in important compartments like the lymph nodes. And what they showed was a combined blockade, both of these together, led to greater virus reactivation as well as increased expression of RNA in CD4 T cells and in B cell follicles. And this isn't compared to PD-1 blockade alone or in controls where the effects were much less substantial. So let's move on to CROI, where Thomas Rasmussen from Sharon Lewin's group presented an interesting reservoir sub-study from AMC 095. So this is a phase 1 study of checkpoint inhibitors in people living with HIV and cancer. And what he presented were data on a subset of individuals where they were able to do reservoir assays. And importantly, in these figures, I'm showing in blue, or he showed in blue, single-agent anti-PD-1 therapy, and then in red or orange, combination PD-1 and CTLA-4 activity. So in the first graph, looking at infectious units per million, or sort of virus outgrowth before and after, or while on checkpoint inhibitors, you can see in our whole group, there's minimal changes sort of in reservoir measures. But if we look at a small subset of individuals who got both PD-1 and CTLA-4 blockade or combination blockade, we can see there's a trend, this is just 2 individuals, but a trend towards more substantial reductions in the size of the reservoir. Further, looking at just HIV RNA expression in dual checkpoint receipt, you can see that there was a trend, or a marginal p-value for increase in expression of RNA. So I think in this this population of people living with HIV who also have oncology indications, these are also exciting data for dual blockade. The authors went to pains, I think, to express that these are exciting, but also to state that the current formulations have real concerns for toxicities, and the hope is that novel formulations or new ways of using, trying to elicit checkpoint inhibitors or blockade could allow these strategies to become more realistic to be used in HIV-positive individuals. All right, so moving on to broadly neutralizing antibodies, which have been a component of all of these combination immunotherapy strategies. So it's been shown for a couple years now, due to work from Michelle Nussensweig and Marina Kasky, that combination BNABs could be effective in suppressing virus in individuals with sensitive reservoirs, as shown in the middle here, whereas if you have resistance in your virus reservoir, they are much less effective. There is also hints that these antibodies might, in rare individuals, and this has to be borne out by much larger studies, maintain some sort of immunomodulatory effect after these antibodies had cleared. So in order for these antibodies, to be used more widely, there are many groups working to advance these antibodies. And there's just multiple platforms that are being worked on in terms of enhancing the breadth, the effector function, the half-life of these antibodies. And right now, as we sit here, although many of them are paused for the COVID epidemic, there are lots and lots of clinical trials looking at combinations of BNABs together, as well as with LRAs and immunomodulators, to sort of really move the BNAB field forward. And I wanted to highlight one of the symposia that was led by Dr. Michael Farzan. So Mike Farzan has been a leader in this field by developing one specific compound called E-CD4IG. And this antibody-like molecule, or immunoadhesin, has the ability to bind both to CD4 and to co-receptor binding sites, and therefore is really, as shown in this third figure in this breadth and potency curve, has really exceptional breadth and potency when we compare it to a lot of our more traditional or natural antibodies. The other thing Mike Farzan and his team have worked on is delivering this antibody by AAV, or adeno-associated vectors, or virus vectors. And this enables, with a single injection, in theory, we could have long-lived production of this antibody or immunoadhesin over weeks to months. So shown here on the right are examples from one specific rhesus macaque, and on the bottom a comparison of their cohorts of controls versus AAV-delivered E-CD4IG. And in general, what you can see is in some of these animals, we're getting very high levels of E-CD4IG, shown in blue. after AAV delivery, and that is leading to virus suppression, as shown by the black line. And so there's a lot of ongoing development in terms of enhancing the delivery, the vector delivery, as well as the half-life of these compounds. But I think this is a very promising way that we're going to be able to deliver antibodies over a longer period of time to a wider breadth of the HIV-positive population. And so I'd encourage you to watch his seminar if you have the time. Another interesting presentation along those lines was by Joseph Casaza of the VRC, the Vaccine Research Center, and he also described a phase 1, an early clinical trial, human clinical trial, using AAV delivery of a BNAb. So this is VRC 603, a study of 8 individuals that were given either low, medium, or high doses of AAV delivered VRC-07. And what you can see on the right, where in red, the graph of VRC-07 concentrations in the plasma is shown in red in the 3 high-dose recipients, is that in some of these individuals, they were able to generate detectable and sustained levels of this antibody over many, many months. Now, there were anti-drug antibodies that developed in some, and that can correlate with some of the loss of detectable levels, but not all. But the point is that this is a first step in using AAV delivery of antibodies in humans, and a very exciting opportunity for us to deliver these things in a longer period of time. And so for my final slide, I wanted to mention another very exciting area of cure research that I didn't have time to go into in much depth, but that's the whole world of gene and cell therapy. And I thought Chris Peterson from the Hutch at the University of Washington gave a lovely symposium describing 2 components of cell and gene therapy and the advances that have been made by his group and others recently, specifically in the preclinical non-human primate model. So on the left, he detailed a really nice iterative process where, using experiments in the non-human primate model, they were able to distinguish what was the most important correlate of the stem cell or the bone marrow transplant cures that have been seen in individuals like the Berlin patient, Timothy Brown, and the London patient and the Dusseldorf patient, and other patients who've achieved durable remission after stem cell transplantation. And what he showed in a series of experiments was that it was the transplant of HIV-resistant stem cells, not the graft-versus-host disease or severe conditioning regimens, that were essential for, or was the key component of that durable remission. So that's something that they're building on in order to try to improve our ability to deliver cure via stem cell transplantation. He also detailed a lot of progress that has been made in CD4 CAR T cells. And this work is to develop these effector cells where you take cells from an individual and you engineer them to be more effective at targeting and killing HIV-infected cells. And so he detailed multiple steps that have been done by himself and other groups to increase the persistence or the ability of these cells to circulate in the host for a longer period of time, to specifically target HIV reservoirs, and then what we need to do to take these sort of boutique medicine approaches and move them forward into scalable approaches that can be used in a large number of individuals. Okay, so in summary, I hope I've had the opportunity to highlight some of the really outstanding reservoirs and cure research that was presented this year at CROI, and hopefully encourage you to look back at the website and pick through some of those lovely presentations. In general, I tried to highlight the fact that advances in understanding the biology of the reservoir in terms of these key questions like, when is the reservoir formed, how is it maintained, what is the role of expanded clones, and what are the key tissue and cell compartments, these are some of the strategies, or some of the advances that have been made. I also discussed advances in reservoir measurement in terms of both imaging and then applications of the intact proviral DNA assay to clinical trials and cohorts. Then we discussed novel latency reversal agents and immunotherapy strategies, which have shown promise both in the level and consistency of latency reversal they've shown in animal models, as well as in combination immunotherapy strategies, both in rhesus macaque and humanized mice models, as well as early phase 1 studies. And then finally, I just want to mention, I barely touched on the fact that there have been impressive advances in cell and gene therapy and the vector delivery of antibodies and other important reagents. These have shown promise in preclinical animal models, and there are exciting phase 1 clinical trials ongoing. So with that, we will move, I believe, to our post-test questions. So the first post-test question is, as the IPDA, or the intact proviral DNA assay, has been applied to cohorts of ART-suppressed people living with HIV, we have learned that A, CD4 T cells with intact and defective provirus decline at the same rate, B, CD4 T cells with intact virus decline faster than those with defective virus, or C, CD4 T cells with intact provirus decline more slowly than those with defective virus. Okay, well, it looks like we have a majority of individuals recognizing that the IPDA assay applied to 2 different distinct cohorts of people living with HIV showed that that cells bearing intact virus declined more quickly than defective virus. And this is really encouraging because it suggests that our immune system is able to recognize cells infected with the real barrier to cure, replication-competent virus, and sort of attacked it more effectively than the defective virus. Okay, great. And then our post-test question 2. This still may be a little bit tricky. I don't know if I emphasized it well enough. Which of the following is false about latency reversal agents? HDAC inhibitors tested in human clinical trials are generally safe but do not lead to increased plasma viremia or decreases in the reservoir. Non-canonical NF-kappa B activators demonstrate both latency reversal, plasma viremia, and reservoir reduction in ART-suppressed non-human primate studies. Or TLR7 agonists in combination with broadly neutralizing antibodies have shown delays in rebound after treatment interruption in ART-suppressed non-human primate studies. Okay, great. Again, we have a winner with B, and the point I was trying to emphasize here is even with our most exciting new LRAs, including the non-canonical NF-kappa B activators, these have not yet shown substantial reservoir reduction. They have shown plasma viremia, and we're going to need to do additional studies pairing them in combination with other strategies or with further modifications in order to see the ultimate goal of reservoir reduction or delay in time to rebound. All right, question and answer center section. Okay. I only see a couple questions, and I'll try to address them at least briefly. So the first question for Arun is this presentation only covering immunotherapy, or will it go over gene therapy? Well, that is a fair critique because I really have emphasized the immunotherapy strategies, and I gave sort of a cursory review of the gene therapy. I really would recommend Chris Peterson's presentation. It's only about 25 minutes. And he gives a lovely description of some of the highlights in some of the gene therapy realms. There were multiple oral abstract and poster sessions describing gene therapy advances, and so I don't mean to give short shrift to that very exciting area of cure research, and there's a lot on the website in that regard. Okay. Regarding cell and gene therapy, all efforts seem to be aimed at developing CCR5 deficient CD4s. How does this address CXCR4 tropic virus? That's a good question. I think a lot of the studies right now there are studies that are primarily looking at CCR5 deletion in terms of cell therapy. I'm aware of other strategies where autologous cells are being modified to be resistant to both CCR5 tropic as well as X4 tropic viruses. There are several other molecules that can be placed on the surface that can block either co-receptor targeting viruses. So I do think that there are multiple strategies looking at those sort of options. All right. If there are no other questions, I will say thank you so much for your time and attention, and I hope you have a wonderful day, and good luck to all of you out there dealing with the COVID-19 pandemic, and I hope you stay safe and well rested. Thank you.
Dr. Barr, there is one more question that has come in. We'll move it over shortly before we turn over.
Okay. Okay, one question was raised. How does the reservoir affect SARS-CoV RNA? I think that the general comments that can be made about how SARS-CoV-2 and coronavirus interacts with HIV are that it is still, I think, a largely unstudied issue. I am not aware of many studies. Even I know there are cohorts and there are collections of people attempting to address clinically how does COVID-19 affect our people living with HIV, our cohorts in the United States. I'm aware of several of those studies, but I am not aware of results from those. In terms of how does SARS-CoV-2 affect the reservoir, I have no data to base this on, but mechanistically, we think that SARS-CoV-2 doesn't necessarily infect the same populations that are the predominant cells of the reservoir, CD4 positive T cells, so there may not be a huge interaction, but I really think all those studies will have to come later, and there's so much to be learned about COVID-19 as well as how it specifically affects cure issues. Another question, how about ART and CRISPR in cure? So there are clearly a lot of, this may be, there have been multiple studies published over the past year looking at laser ART and CRISPR technologies, and this is really a very active field. And last year in my webinar, I covered this in a lot more depth, and I just, because there's so much information, didn't spend a lot of time on it here. But I wouldn't, this is a bit of a dodge to your question, but I also think there's a lot on the webinar in terms of CRISPR if you're looking at recent advances that would be very interesting for you. Okay, Donna and Jose, any other questions? Or are we?
No, I think we're done. And everyone, thank you so much, Dr. Barr, for a fantastic review of what was presented at CROI. I'm checking that Jose is on.
Yes, and I'll go over the remainder of the closing slides.
Okay, terrific. Thank you. And thank you, everybody, for attending this webinar. We'll go through some housekeeping remarks, move over to Jose.
This webinar will be available on-demand viewing and CME credits at the IAS-USA website. CME credit cannot be claimed for both live and archived sessions of the webinar. As a reminder, evaluations will be emailed tomorrow by 5 p.m. Pacific time.
And I think we sent a note to everybody just as a reminder. We have to process each one of your records in order to record that you participated, and that takes time. That's why we can't just automatically have your record updated, because we have to go through each of the 400 people or so who signed up for the webinar and make sure your records reflect that.
Due to the COVID-19 pandemic, our live spring courses are being postponed. For the latest updates on postponed activities, please visit the IAS-USA website. We have robust programming planned for 2020, and the next webinar in our webinar programming will be tomorrow, titled Wellbeing During COVID-19 Crisis for Healthcare Practitioners. The registration for that webinar will be closing today at 4 p.m. Pacific time. So if you have not yet registered, we encourage you to join us in this webinar programming. We would like